The Ag-West Boffins Innovation Series is about promoting innovation in agriculture, and it's about bringing together the people with those innovative ideas with the end users for those ideas, whether they be business or actual people who will use it on their farm or in their business. So it is really about connecting people and connecting and growing research and business here in Saskatchewan. So this afternoon, we are putting the spotlight on genomics and growing your business with genomics technology. And specifically, we'd like to highlight the Genome 360 project, which is administered by our partners today, Genome Prairie. My name is Karen Churchill. I am the CEO and president of Ag-West Bio. So I am your MC for this event and happy to be so. And I'd like to acknowledge that today we are on Treaty 6 territory and the homeland of the Métis. We pay our respects to the First Nations and Métis ancestors of this place and reaffirm our relationships with one another. I'd like to especially thank the sponsors of this event. They are the Global Institute for Food Security, Energene, and the Crop Development Center. I'll move now to introduce our first speaker, which is Lizzie Mascarenhas. She is I hope I haven't slaughtered your last name. She's the Chief Scientific Officer for Genome Prairie, and many of you will have known her. She's actually a former Ag-West Bio employee. She's worked for SaskPulse. She knows our industry very well. So welcome, Lizzie, to the podium and to introduce Genome 360.
Good evening, everyone. I feel like I'm amongst friends today, so I'm not going to say a whole lot about me, but wanted to say how thankful I am that Karen was kind enough to accommodate us and with her wonderful team that has supported us through preparing for this event as well as launching the Genome 360 officially. We had one slide. I don't know where Rahwa is. Have you got a slide for me? So thank you, Karen, for inviting us to talk about Genome 360 today. While that slide's getting ready, all I'll say is Genome 360 is a program that is that has been launched by Genome Prairie over the past couple of years, and the bigger events and the bigger footprint we've had has mostly been in Manitoba, where Genome 360 has had tremendous impact on the Cancer Care Manitoba genomics suite capacity building, as well as in some of the programs that we've done on the HQP side. Over the last year and a bit, we've launched Genome 360 Saskatchewan, and it really is about three things. It's about people, opportunities, and solutions. So that's what Genome 360 is. In both provinces, it's meant to create the environment and facilitate and support activities and partnerships that will create value and add value to the agriculture, health, and natural resources sectors, all enabling through genomics. Solutions, as I mentioned, are for challenges that any business might have. We are interested in partnering with like-minded entities in the region to build solutions for the challenges that public and private sector partners might have. We're interested in looking at new opportunities that result in new products, service, or processes that would improve efficiency, speed, and accuracy. So the promise of genomics is all about doing things faster, better, cheaper, and more efficient. That's the promise of genomics. And finally, the last one is, but not certainly the least, is about building capacity in the province. We have a lot of potential, still untapped potential. So we're hoping that through the Genome 360 program in both provinces, we'd be able to build a lot of HQP that focuses on enabling more of the solutions and opportunities to be built in both provinces. So with that, a short introduction about Genome 360, I'll pass this off to and I have to say, I wanted to introduce the two people, the two stars that I have in my team that are actually working on making this a reality in the province of Manitoba is Raquel Bert and Rahwa Osman, who is in Saskatchewan. So now I'm going to pass this off to Rahwa, who is going to speak about her plan for Genome 360 Sask. Thank you.
Thank you, Lizzie. Thank you, everybody, for coming. I know it's not exactly an ideal time in life. So let's talk about Genome 360. The general idea here is we're looking at adopting genomics away, not so much as a way, when people think about genomics, they think research, they think VIDO if we're in Saskatchewan, they think of University of Saskatchewan. What we're looking at is trying to use a product of research, if you will, to everybody's everyday life. Answer questions, apply it in our food, livestock, water quality. It is to make it useful and accessible to everybody, the general public, SMEs, to build our economy with it. So to make it very useful away from just theoretical. So with Genome 360, our goal is, like I said, make genomics accessible and useful. And the way we're going to go about it is we're going to do it through business community engagement. So we're going to do it through our businesses, so our businesses could be genomic testing, they could be service giving, they could be people who are end users of genomics. But right now our focus with this project, with this program, is let's adopt it into our business community. And it's just not chit chat, it's also to find solutions through it. And it's tell us what your challenges are. We'll see if genomics would actually be the source of your solution. And that is essentially behind our Grow Your Business Through Genomics slogan. And the way we're going to do it is we're going to assess your need, we're going to facilitate solution, and essentially we're the matchmakers. We're going, if you need services through genomics, we would look at what services are available to you that within the province. So, and the next thing is, we reduce it to practice and then assist with deployment. So that would mean we can build an infrastructure, we can build an HQP training, we would need to build a workforce to actually make it happen. And the next thing is, we want to give industry services. That is specifically important because instead of sending it to Manitoba or Alberta or even out of the country, now we can actually do it within our province. And more importantly, we see this now during COVID times when borders are closed. You want to be able to go drop off your sample, to drive over to Regina to do it. So it keeps life going if we have the capacity within the province. And why? Well, it's to find solutions to challenges essentially. And it is also to contribute to the economic sectors. So overall, we're trying to add value with genomics. And we're trying to apply cutting edge technology, which is already proven in different areas. And you'll hear that from our speakers. There would be Carl Potts from the crop industry. There would be Dr. Cheryl Waldner from livestock. So there are examples where they've been applied in Saskatchewan. But in the end, focusing on agribusiness, our target audience here is Saskatchewan-based SMEs. We're providing solutions to challenges, and the way we're going to do that is we're going to look at improving production. So you can ask, okay, yeah, I want to improve my production, but how am I going to do it, right? So if we're going to look at, say, in this case, milk production, how genomics fits in is you're not going to be sequencing every day, but what you do is you look at preferred traits through sequencing so that when you're actually buying a herd, for example, for milk production, you would select for a preferred trait applying genomics. So in addition to making business decisions, it moves our practice forward. We bring it to the 21st century, if you will. And the other thing is disease and insect resistance. So this could be, maybe you're using a lot of pesticides, but now if we actually find a resistant trait, you'd be using less. It's environmentally friendly. It might even be cheaper. And for more important things like the climate resilience now, we have seen a heat wave this summer, and I'm sure the canola growers would have loved to have, heat resistant strains. And the way we find out is to look at the genetic traits. And in terms of reducing losses, we're also looking at what causes losses. And so let's say it's fungus. So we actually screen what we're going to be storing to avoid this fungus being in that, so that it actually ends up creating spoilage. So that's basically some of the examples, but you'd hear a lot more from our speakers today. And in the end, if you're asking how Genome 360 comes into play, we're like I said, we're the matchmakers, and you can contact us about the opportunities. If you're wondering whether there's any chance if you could use genomics, call us. I can give you the technical advice. We can talk about this and see if you should continue doing what you're doing and that's the best way for you, or you can actually manage to adopt genomics to make it faster and cheaper. So in the end, call us. So for everybody here and everybody in Saskatchewan, call me. I'm Rahwa Osman for Saskatchewan. If you're in Manitoba, that would be Raquel. Contact her. If not, just contact any one of us. We can have, this is just a chat away. With that, I would like to introduce our first speaker that is very kind to give us some of his time. This is Carl Potts. He's the Executive Director of Saskatchewan Pulse Growers, and he's been in this role for 10 years. Prior to joining SPG, Carl led market access, trade policy, and international market development work for Canada's National Pulse Industry Association. Oh, that's a mouthful. And Pulse Canada. He's a graduate of University of Saskatchewan, College of Agriculture and Bioresource. Please help me welcome Carl.
Thanks, Rahwa, for the introduction, and thanks to Genome Prairie and Ag-West Bio for the invitation to speak here today. I do have a presentation with a few slides that I want to work through, and I know some of my colleagues that are speaking a little bit later, have deep expertise on the science side. What I hope to bring here today was talk a little bit about the business side and the application of genomics as we see it in the pulse sector here in Canada. So just quickly, who are Saskatchewan Pulse Growers? We're a development board for the pulse crop industry here in Saskatchewan, and we represent about 15,000 grain producers who produce pulses here in the province. We're funded through a checkoff or a levy. Most of our funding comes from directly from producers through this checkoff or levy, and that's paid on the sale of pulses when farmers sell grain to grain companies. So that's basically who we are and how we're funded. But what do? We primarily make investments into research and market development, and we advocate on behalf of the pulse sector here in the province. So at SPG, we have a very heavy focus on funding and supporting R&D. And I'll talk a little bit about some of the impacts of that a little bit later. I'd like to give a bit of a snapshot of growth. And I think the story of pulses in Saskatchewan is really one of growth over the last 30 years. From this chart you see on the screen, from the early 1990s to now, we've had very strong growth in both the quantity of production as well as the seeded area of pulses across Canada. So from under 1 million tons of production of pulses in Canada in the early 1990s to up over 8 million tons of production in 2016. So the blue bars are tons, the red line is seeded area. And as you can see, production is increasing at a faster rate than seeded area. And that really signals advancements in productivity, really due to enhancement of varieties, better technology that farmers are adopting, and as farmers become better at producing these crops. So the story here that I just want to drive home, is about growth for the pulse sector in Canada. If we look specifically at Saskatchewan, this shows seeded area in acres for Saskatchewan for the various pulse crops. Again, very much a story of growth. The red portions of the bars there are peas, the blue bars are lentils. And as you can see, in about the mid-2000s, 2006, 2007 or so, really strong ramp up in seeded area of lentils. The introduction of new red lentil varieties for farmers really expanded that adapted area of lentils in the province and has really grown since then. If we look at Saskatchewan's share of Canadian pulse production, Saskatchewan's very important in the Canadian context, in the local context, provincial context, as well as in the international setting as well. So Saskatchewan would represent about 50% of Canada's pea and faba bean production, and we produce over 90% of Canada's lentil and chickpeas right here in Saskatchewan. So I want to talk a little bit about why genomics. Why is genomics important to farmers? So most of the gains that we've made to date, we believe have really been through traditional breeding techniques. So a lot of the gains so far have been through those sort of techniques, but the problems and the opportunities are becoming more complex. Trying to make advancements in yield gains, diseases, weed control, improving end-use quality. All of those factors are becoming more complex and harder to achieve as the crops become more developed over time. So genomics is really one of those tools that provides the possibility to achieve greater results faster. And I think Lizzie mentioned some of that in her comments before. So why do we as a growers organization make investments in genomics? We're trying to make those larger advancements in varieties more quickly. We're trying to advance yields. The question we often ask is, can we make the breakthroughs in pulses like has been done in the canola sector with the creation of hybrids, for example? So how do we make those stepwise jumps in yield to help ensure producers remain competitive and productive over time? Genomics helps with the specific selection of desired traits with reduced time on phenotyping, to develop improved end-use quality. So things like protein content, flour and protein functionality are particularly important as end-use quality characteristics for pulses for markets of today, but especially for markets of the future. And lastly, to better withstand pest pressures. So really, why is this important? All of it is really trying to drive towards more competitiveness and enhancing that competitiveness, not only with competitors internationally, but as we compete with advancements for other crops like canola, cereal grains, and other crops that farmers produce in Western Canada. I thought I'd touch a little bit on what we've invested in to date. So our investments in genomics date back at least to 10 years to 2011 or so, with some initial lentil genomics research. In 2013, we made some major investments along with other co-funding partners into lentil and pea genome sequencing projects. And a lot of that work, really all of this work, has been led or had a very significant component done here at the University of Saskatchewan Crop Development Center. 2014 to 18, some focus on trade associations, yield and climate resilience traits, resequencing of chickpea lines, as well as tool development is sort of some of the projects that we're involved with right now on chickpea. But the couple I've left to the end here are two large Genome Canada projects that we've been major supporters of, along with many others. First one is the Agile project that ran 2015 to 2020 or so, led by Dr. Kirsten Bett from the University of Saskatchewan, as well as an additional project starting in about 2019, the Evolves project on lentils. So we've been involved in providing support, financial support and funding for both of those initiatives, really to help advance genomics, advance some of these objectives on lentils. So if we do some rough math and kind of add some of that up, about $9 million worth of investment over the last 10 years or so by farmers through SPG in support of genomics related projects. And we think that that's going to be really key investments for producers' competitiveness as we go forward. So just finally, where are we now? So, even though we've been at some of this for 10 years, we feel we are still in some ways at early stages and at that tool development stage. And so now it's about translating, that knowledge of genomics into varieties and actually, delivering those in terms of improved traits, improve variety performance for producers. So some of the things that are being worked on, chickpea flowering time response for maturity, improve resistance to Ascochyta. On peas, Aphanomyces, which is an important root disease that's really impacting our sector here in Western Canada, as well as protein content are important areas of focus. And on lentil, disease resistance, additional herbicide tolerances, and end-use quality are all very important traits for farmers that we really hope and feel genomics can help unlock some of that potential. So at the end of the day, we make those investments, to benefit growers. It's very, from our perspective, it's very applied research with very specific end goals in mind, and we're excited about what some of those opportunities will be for the future. So with that, with just 10 minutes, I thought I would end there.
Thank you, Carl. We're going to continue on talking about crops, and the next speaker is Sean Walkowiak. I am so sorry, Sean. And he comes from the Canadian Grain Commission. He is a research scientist and program manager for microbiology and grain genomics. And he's also an adjunct professor at the University of Manitoba in the Department of Plant Sciences. Sean's teams focus on applied research for disease and crop surveillance, as well as disease management, with a focus on Fusarium head blight disease of wheat. And his team participates in a number of national and international projects that use genomics for wheat improvement, including the 10-week genomes project and the 4D wheat. So welcome, Sean.
Yeah, thank you. I'm really excited to be talking to you guys today about genomics. Why now for genomics in agriculture? So I think if you look at historically, there hasn't been a lot of activity in genomics, especially in crops, and it largely had to do with the size and complexity of these genomes. So right now, it costs about $600 to sequence a human genome, whereas if you go back 20 years when they started sequencing the genome for humans, it would cost over 100 million. And the same is true for wheat, which is actually, much larger than even the human genome. So there was some early progress in genomics for, bacteria and fungi, with the Fusarium genome being sequenced in 2002, and I'll talk about Fusarium in a second. But only recently have we really gotten to these tools for wheat, and a lot of it has to do with the cost, and the technologies just simply weren't there, but they are now. So now is the time to move on it for crops. So if you look at the priorities for crop improvement, there's yield stability, trying to protect yields against losses from things like fungi and insects, and also trying to improve the marketability of our crops. So for example, protecting against Fusarium head blight, which is a disease of wheat, which produces toxins that affect producers and how much they can get for their grain. For end-use quality and marketability, for durum wheat, yellow pigment for making nice yellow pasta, low metal content in the grain. For protecting crops again, resistance to insects, this is the wheat stem sawfly, which is conferred by this trait here with a solid stem. And all of these traits, the underlying basis of them is the genetics. So using genomics, we can now get access to this information and apply it in breeding. So the way that this looks like, I've sort of represented the breeding cycle here in purple and research here in blue, and they both sort of feed into each other at every stage of the process. So, breeding starts with selecting the right parents with the right genes. So understanding the genomics of those parents helps you better select those individuals for crossing. Then when you're advancing your populations in breeding, you need to test them for the genes to make sure that they carry them. And at the end, you want to see, okay, now that they carry the genes, is the crop behaving as I would expect based on its genetics? Is the biological system working? And if it's not, it feeds back into research, identifying new genes, selecting new parents based on their diversity. So it's really applied at multiple stages in the wheat crop improvement pipeline. So towards this, we had the 10 Wheat Genomes Project, which looked to develop high quality reference genomes for different wheat lines, including Canadian wheat lines, CDC Stanley and CDC Landmark, that were developed at the Crop Development Center. So we developed the genomes for these lines here, and they only came available last year. So this is all groundbreaking new stuff because of the advancement in those technologies. And we can use, information from other wheat lines from around the world at different levels of sequencing, different levels of genomic information, to really get at as much diversity as possible so that we can apply it in a breeding to develop the best crosses and material possible. So it's not just about generating data, it's making the data available. So shown here are some different tools that were developed from our projects, where we really wanted to get at the genes and the diversity and make it available to general users that can look and compare the genomes, identify the genes, and compare them between the different wheat lines to help make important decisions for breeding. So I'm going to switch gears a little bit and talk about Fusarium head blight, which is an important disease, especially in Canada. So in 2016, we actually had an epidemic year for Fusarium head blight. And I think the estimates were maybe around $1 billion in losses, primary and secondary losses caused by the disease just in 2016 alone. So it's a really big deal. This is what the grain looks like when it's healthy. This is, different degrees of damage. You can see, it's been spreading throughout the prairies and has been increasing in prevalence over the last few years. So the Fusarium genome was so important. It was one of the first ones to be sequenced in the early 2000s. And you can see the amount of research productivity that increased just from the availability of the genome. So the genome became available around here, and then all of a sudden it's stimulating all this research and information that can be used to understand and control this pathogen. So to give you an example of the way that we use this information, so these are some of the technicians in my lab, and before genomics, the way that we would identify the different species that are causing this disease, which is a fungal disease, is we'd grow the fungus. So that takes a few days, and then you have to put it on some microscope slides, look at it under a microscope, and then you have to, look under the microscope, identify the different shapes and morphologies of the spore to give it a proper ID. And you could do, a few hundred of these before going cross-eyed under the microscope, but it's quite tedious and it's a lot of work. So if you look at what we do now, we have high throughput genomics testing. So we can use robotics to do our auto sampling of hundreds of samples all at once. We can test them for many different things in these robotic systems. So here there's a chip, and that chip has 5,200 little holes in it, and we can do 5,200 tests on one chip for hundreds of samples, testing for all sorts of different things. So here we test for the different species using markers that we've developed by comparing the genomes of the fungi. You could also test for things that you can't see under the microscope at all, things like what types of toxins these fungi are producing. So to give you an idea of the throughput, over the last few years, we've ran almost 40,000 samples, and on each one of those, we've run 24 different tests. So we're looking at, over 800,000 different DNA tests we've run on these samples, something that you could never do under a microscope. So it just massively increases your throughput and ability to screen for these diseases so that we can, help keep the industry one step ahead through surveillance. So to summarize what I've talked about, genomics is important so we can capture the diversity, both on the crop end, on pathogen end. You can use it to develop tests that you can use to monitor, lines and breeding programs, monitoring pathogens as part of surveillance program to help support the industry. And it's really an exciting time to be in genomics, especially now, given that the cost of the technologies has changed, and there's all these new available technologies that can really massively increase your throughput. So a lot of these projects are very collaborative. The 10 Genomes project, for example, had over 100,000 or 100 authors on that project, as well as the collaborators involved. And many of those were students, there were postdocs, So, trained a lot of high quality personnel. A lot of these were led in Canada, supported by Genome Canada and Genome Prairie, and really helping build our capacity and resources to be able to do genomics here in Canada. So it's great that we're leaders in that. And that project was led by Curtis Posniak here at the University of Saskatchewan. So yeah, thank you for inviting me. And yeah, it was exciting to talk to you here today.
Thank you, Sean. Next, we switch gears to the livestock industry. It would be Dr. Cheryl Waldner, who was my personal teacher at One Health, and she's absolutely amazing at what she does. But more to her attributes, she is a professor at the Western College of Veterinary Medicine, also known as WCVM, and an NSARC Beef Cattle Research Council research chair. She is particularly interested in antimicrobial use and resistance, also randomly known as AMR. I'm sure you've heard it around. She is an expert in that, and as well as the prevention and control of infectious diseases. Cheryl?
Good afternoon, everybody, and thanks for having me. I'd like to talk today about an example of how we're using genomics in the livestock industry. Let's try to get my system to cooperate here. All right, come on. Sorry, that's really bizarre. Anyway, hopefully that will work. All right, so the example that I'm going to talk about is bovine respiratory disease in the beef industry. The beef industry is Canada's largest livestock industry. We have exports of more than $3.7 billion a year. And this is something that impacts probably the most farms and farm families across the country when we look at the livestock industry as a whole. We still have, even with consolidation, we still have more than 60,000 farms and more than 200,000 jobs, and then spin-off jobs from that. So this is a very important industry. Bovine respiratory disease is the single biggest disease threat to that industry. And we spend close to $50 million a year just in treatment and control alone. Respiratory disease is a really timely topic right now. We're dealing with COVID, and this isn't much different, but it is a little bit. We do see the same effect that we're seeing right now, where we've got a spike that's to at least some extent associated with kids going back to school. Calves entering feedlot pens are very much like kids in a classroom. We get these highly mixed groups of animals from different sources. They're stressed by recent weaning and transportation, new surroundings. They don't know where the food is. They don't know where the water is. The vaccination status of these calves is unknown in many cases. And that leaves them very, very susceptible to disease. So the difference with what we see in a lot of respiratory disease in people is that a lot of this is actually, while it might be triggered by a virus, it does lead to bacterial infections. And those bacterial infections do need to be treated with antimicrobials. So to get the right antimicrobial, we do need laboratory testing that can help us figure out which drug we need to use, which animals which we need to use it on, and what's the best timing for that. And right now, almost 92% of feedlot cattle actually receive an injectable antimicrobial for either the treatment or management of bovine respiratory disease. So this is a big issue. And it's an even bigger issue because antimicrobial use in veterinary medicine is coming under increasing scrutiny. The World Health Organization a few years ago issued some guidelines that strongly suggested that antimicrobials that are critically important for human medicine shouldn't be used except when we can show using a diagnostic test, and they singled out bacterial culture and sensitivity, that drug was actually necessary and would be effective in that particular case. This policy is supported by McDonald's and their global vision for antibiotic stewardship. The problem is that culture and sensitivity can take up to 5 to 7 days. So what have we been doing in the livestock industry? Well, current best practices are as good or better than what I've seen in a lot of human hospitals. We have a really strong regional lab at Prairie Diagnostics here. They're using a multi-TOF machine, which is functions using lasers to help us identify the specific bacteria, but we still have to grow them and we have to wait for them to grow. And then we need to grow them again to test whether or not they're sensitive to specific antibiotics using sensititer technology, which is what is used at the national level for the surveillance programs for both human and animal health. So we are using the currently available best practices. But again, this whole process takes 5 to 7 days. We have to collect the sample. When this is being done in the field, it's got to go through the courier to get to the lab. That can be 24 hours. What we've got to hear on the thing is highly optimistic in my experience. Usually it's longer than that. And we got to grow it up for 24 to 48 hours. And then we need another 24 to 48 hours to get our sensitizer results and then get the report back to the veterinarian and to the livestock owner. Well, that's just too long. So we need to do better. So we need something that's faster, we need something that's cost efficient, and we need something that's going to inform practical guidelines for disease management. This sounds a lot like the genomics commercial that we were hearing in the previous two talks. Well, what we're focusing on at the Western College of Veterinary Medicine is metagenomic whole genome sequencing. This suite of techniques allows us to streamline a lot of laboratory processes we used to do separately. And I'll talk a bit more about that in a minute. It has been shown to demonstrate a lot of value in the human healthcare field. There's many countries in the world that have switched a lot of their human health diagnostics almost specifically to genomics. It's much more standardizable than a lot of the other techniques that we're dealing with. It's safer for the people in the lab because we don't need live pathogens. We just need the DNA or RNA. And there's so much information that can come out of a single test. We can figure out what the bugs are. We can figure out how related they are to each other to try to figure out transmission chains in terms of where did the bugs come from and do outbreak investigations. We can look at the sensitivity to specific drugs. We can look at how virulent or how scary they actually are. And we can look at whether or not we've got a new emerging bug from a particular infection threat, or whether it's something that we've known about for a long time, and allow us to sort of, if there is an emerging bug, allow us to track the evolution of that over time. So just to get into this a little bit more, what we're doing with these techniques is we're directly testing the sample itself, which takes out this culture and sensitivity step and makes it faster. We can characterize all of the DNA or RNA in the sample, allowing us to basically cast a net for everything that is there. We can look at all of the pathogens that are there, whether they're bacteria or viruses and all the different types of bacteria. We don't have to know ahead of time what we're looking for. So we can look for both known and previously unreported pathogens. So this is really important because it doesn't require us to optimize the test for every single target of interest. We don't have to have a list of things that we're looking for specifically and then spend, a lot of time and money trying to optimize the targets, which a lot of the current molecular techniques sort of limit us. And PCR is one that you might have heard of if you're in the livestock field at all. It's a great tool, but we need to know what we're looking for. We're not going to find it if we don't know what we're looking for. It really does give us the potential to develop this idea of one test for all. There are some challenges when we're doing this type of direct testing. There's a lot of noise that's created by the DNA from the animals themselves, and we've got to figure out a way to best filter that signal that we're interested in out of the noise. So again, just to kind of pull this together, this whole idea of this next generation sequencing or this third generation sequencing is that we're able to kind of use a net to scoop out all of the information in the sample rather than just casting a single hook and hoping that we're going to get the thing that we're looking for. The other advantage of the newer methods that we're dealing with right now, so again, why now, right? Well, we've got a whole suite of new techniques, this whole genome metagenomic sequencing that allows us to look at the whole story. The example on the slide here is, if we were looking at Canada, if we were looking at Canada, where we're actually going to get the lines to Canada in our results. With the older based type of sequencing techniques that we used to use, we would just get parts of phrases, not even sensical parts of phrases in many cases. And we would get a whole bunch of copies of these, and then we would have to assemble them and hope that we could make sense out of what we had. So it gives us a much more direct line to useful information. The other thing it does is gives us flexibility in terms of how deep we need to dig and how much money we need to spend. We can spend a lot, we can look in a lot of detail if we're trying to look at a new pathogen for the first time and kind of pull the pieces together. But we can also work with data that is much higher level data, sort of the map versus the Google Earth view of the situation, if we just want to create family trees out of the data, or if we just want to do diagnostics to figure out whether or not we're actually seeing something in our sample, a specific set of targets of interest, or anything that might be a target of interest, whether that's bacteria, antimicrobial resistance indicators, or viruses. So this next generation sequencing that we're working with the most in our lab is nanopore technologies, and I don't know if you can, hopefully you can see the video here, and basically it's a series of pores that allows the DNA to pass through the pore. When it passes through the pore, we get an electrical signal, or a disruption, I should say, to an electrical signal that's going through the membrane, and that tells us what the DNA is. And the beauty of this is we can run it on big machines in the lab, and we can do it very efficiently, but we can also do it on a nice little handheld machine that is something that we can take out to the field. So we're not that far away in terms of diagnostics of, being up there with Mr. Spock and being able to take our handheld scanner out and kind of figure out what's going on out there, which is, as far as I'm concerned, is kind of cool. So what are our current challenges? Well, when we're looking at antimicrobial resistance, which is my focus, our big issues are the time delay to the lab results, the cost for processing these lab results, and the collection of the material in the first place. And genomics helps us with the first two, both in terms of the cost of an individual test, but also options for pooling samples, for looking at groups of animals instead of just individual animals. It allows us some additional options in terms of timing of sample collection. And then we, as part of our work, are not just working on the tests themselves, but the strategies for applying these tests to try to get the best possible information out of them. So just in summary, the one big project that we're working with Genome Prairie right now is called Genomic Assets. This, again, is very intended to support antimicrobial stewardship. We collect nasal swabs from calves, and here's one of my PhD students, Jen, collecting a swab. We're using nanopore technology. We're also comparing it to another rapid technology, recombinase polymerase amplification. This allows us to look at both bacteria and viruses. With the bacteria, it can help us decide whether or not we've got the right antimicrobial for the job. With the viruses, whether or not we have a vaccine gap. So part of our project this year is focused on vaccine gaps, and that's coming with support from Saskatchewan Agriculture and the Beef Cattle Research Council. So we actually got our first pen of calves this week, or today, I should say, for this fall. This is a livestock and forage center of excellence, just east of Saskatoon here. We're going to have 8 pens of 100 calves again. We sample them several times, and then we submit the samples for the traditional testing, this new RPA test in our nanopore metagenomics, and we compare the three. Just to give you an idea of how much of a nuisance it is to get these samples, this little calf is holding pretty still here. And I'm sure any of you have had a COVID test. If somebody came at you with a swab that was that big, you'd probably be cringing a little bit, but these little guys don't seem to mind it too much. then the next part is how do we actually deal with the data? And we've developed some really nice simulation models that actually simulate individual calves within feedlot pens and then the bacteria and the resistome within these calves. So the antimicrobial resistance genes within these calves are depicted in this model. And let's just take a look at this. We can see in the model here, we've got pens filling up. As they go through the model, they go through different treatments. The pens start to empty out, and then the mechanism behind it, we actually have a system of tracking and enforcing rules about what the pens are treated with. Then we can look at what happens to respiratory disease in these pens, what happens to resistance in these pens, and what happens to different types of lameness in these pens. So the key objectives of this feedlot model are to optimize pen level testing. What's the diagnostic, not just the test, but the strategy that gives us the best view as to what's going on. And then what time should we best collect these samples from these animals? How many animals per pen do we need to sample? And whether or not there's the potential to actually pool these samples to save money. So we've got lots of different aspects, not just of the test itself, but the economics of the strategy that we're applying to try to make this practical. And a big part of this project is actually working directly with the big feedlots and big feedlot veterinarians in Western Canada to make sure that what we come up with will actually work for them. So just to wrap up here, in terms of benefits for industry, we're looking at new tools to support antimicrobial stewardship, but diagnostics as a whole. We want to enhance the sustainability of the industry because antimicrobial use and antimicrobial resistance are important for animal welfare, but they've also been identified as key research and key sustainability priorities by the beef industry in Canada. And there also is applications to all animal agriculture. So things like the sampling strategy, the genomics, the bioinformatics tools we're developing, and the risk assessment tools we're developing have the potential for applications in swine, poultry, and dairy as well. And then finally, this really is a timely topic because of the WHO guidelines that I already mentioned, but also the European Union is really starting to look at trade restrictions based on antimicrobial use, and that's something that we're going to have to be very aware of going forward. There's a whole bunch of other current examples if you start to sort of look at this topic on the internet. One of my favorite was the development of targeted vaccines using RNA technology for the swine industry, and Merck Animal Health has actually done a bunch of work in this space here lately. So with that, I'll wrap that up. Just want to acknowledge all of our sponsors for genomic assets and our team.
Thank you. So at this time, I'd love to take a few questions from the audience, or remember there are, if you have a question, just hold the button down on the microphone on your table and we can address it. And if you are online, don't hesitate to type your question into the chat. Who would like to bravely be first? Any takers on questions? Because if you get me started, I never stop. And then I don't notice that there's other people. So, all right, I will give everybody a chance to warm up, and I'll direct my first question at Chris, if that's okay, Chris. You mentioned that there's been this, almost exponential growth in research productivity since you've mapped the genome of wheat, I believe. Could you tell me, how do you describe, what's your definition of research productivity? Is that Sean? Sorry.
Yeah, that might be me. Yeah, so I had a graph showing, this increase in productivity for research on Fusarium after the genome came out in the early 2000s. And that graph was just the number of published papers. But I was around working on Fusarium at that time. And I can tell you that the number of people working on Fusarium has changed dramatically. And a lot of it, people were attracted to work on the disease because it had an available genome, it really unlocked the genes to the researchers so that they could study it, identify what, the toxins and things that are being produced by the fungi. And that wouldn't have been possible if those genome sequences weren't available. So that graph was the number of published papers with Fusarium graminearum in the title through a web search. But having experienced it myself, it was day and night from having a genome and not having a genome in terms of being able to do research on the organism.
Would you like to comment on that, how much of that research has actually made it into best management practices in the growers' hands?
So I'd say a lot of it. When I search for Fusarium head blight in the title of an article, it's about QTL development and breeding, marker development. It's about monitoring of the pathogens in fields. It's about comparing the pathogens in Canada to other countries to see if we're having the same problems that they are and what the differences are. So I'd say it's translated from not just surveillance but breeding, development of fungicides. Having the genome sequence really allows you to tackle this organism from all angles. So I could, if you just do a search for Fusarium graminearum in the research channels, you'll see that, a lot of it's, it's not just research, it's applied. It's breeding, it's development of fungicide, it's management practices. Yeah.
Thank you. I can also ask the same thing of Carl. So you've invested all this money, Genome Prairie, Genome Canada have supported those programs. How much of it is actually ending up in the growers' hands today?
Well, that's a good question. And I think, as I mentioned in my presentation, I think we're partway through that development process. So it really is for us about, how we take that research and improve traits, into new varieties. So an example I can give is I know that there's work being done at the University of Saskatchewan to develop improved Phanomyces resistance in peas, for example. So we don't yet have varieties released to farmers that have that improved resistance, but that is it is under development. So I think we've yet to sort of get to that stage where we're translating that directly to farmers, but we're hopeful that that's coming in the very near future.
Okay, for Cheryl and Lizzie and Rahwa, and forgive me if I missed it and you explained it. I again see, a lot of outcomes in research, and you described the 360 program as providing a matchmaking service. So I'm kind of wondering how the Genome 360 project actually will directly lead to addressing the challenges that you're facing with your livestock improvement programs.
Cheryl, do you want to start?
Well, with Genome 360 specifically, I guess I think there's a lot of opportunity to hook end users up with researchers. But in terms of direct improvements, we are working directly. Our main partner for this project is Prairie Diagnostic Services, which is the main laboratory for Western Canada. And what we learn in real time is being incorporated into the services that they provide for producers and for veterinarians. So we are seeing real-time changes in what the options are for diagnostics, and that has changed very quickly here just in the last couple of years alone. So this is something that's almost changing on a half year by half year basis in terms of what options are available to the industry.
Would you like to add to that, Lizzie or Rahwa?
So all I'll add is Cheryl is coming up with a solution. We'll need an intermediary that is going to take it to market to the end user. So the Genome 360 program is all about trying to create the environment and partnerships and the resources to move this from the research continuum all the way to the implementation piece. That's what the Genome 360 focus is all about.
Can you use your mic that's right there? Thanks, just push the button.
So the only thing I want to add to what Lizzie just said is some of those times it's the fact that you don't know the services exist. So that is where we actually also excel in that, in bringing everybody together, where some of the genomic testing exists, but the end user does not know about it because it's really in the early stages. It may be a startup. So that is how the matching service would actually help.
I know we're running very quickly out of time. It's amazing, but I would like to take a couple questions or 1 or 2 questions from the chat line. So how many businesses so far are linked through Genome Canada, Genome 360, playing matchmaking with the U of S or NRC or other government supported agencies? So that's pretty specific. How many?
Genome 360 is not a Genome Canada program. It is a Genome Prairie program. So the link between Genome Canada and Genome 360 is to look at all the Genome Canada funded projects to see if there are implementable solutions at the end of each project, to see if we would take those solutions to the end users. That would be the link. As to the question that your viewer just asked, I don't have the numbers at hand. I can find that out and pass that on. Yes.
And along the same vein in my mind is there's a question about, do you have a published, so you have bought some equipment and it's at SAS Poly. Do you have a published price list?
No, we're still working that out. So there isn't a lot of equipment at SAS Poly. It is spread throughout Manitoba and Saskatchewan, but there is enough to undertake sequencing and informatics analysis. The question is to, first of all, define what the problem is for the person. And each of the cost and the timelines will depend on the availability of resources already in the public domain versus actually coming up with a plan. So.
Well, thank you.